like to welcome everybody this afternoon to the thesis presentation of David White. Tell you a little bit about David. David is a local person, and he went to school, did his undergraduate here at JSU, and decided he wanted to stay around and saw some of the work that we had been doing, that Greg Skull, one of our former graduate students who's here today, some of the work he did, and some of my first grad students that got me out in the field and started me on this adventure of doing more field work than lab work. But anyways, David was real excited about getting out and really taking on a project, a pretty large project that we had started several years ago, again with some other students, and I think at this point we've I'll let him tell you his story about all the work that he's done. But he's done quite a bit of work, and he's been able to work with quite a few different agencies. Greg now works for Fish and Wildlife, and David's been able to go out and do some work with Fish and Wildlife folks, and some of these other agencies that he's hoping one day to get a job with. But he's done a lot of work, and right now we're compiling this work and some other work that has been done, and hopefully we're going to get this published, get this information out there, because I think he's got a pretty good story to tell. So with that being said, I'll let David tell you his story.
All right, like I said, my name's David. Everybody in this room knows me, or you probably wouldn't be here. So pretty much the idea behind this was we were wanting to know the fish that were in Choccolocco Creek. That's what this is basically based on. We're emphasizing on its greatest conservation needs species, which are a little bit endangered or threatened, and that was the basis of this. So starting out, a little bit of backstory. Alabama waters. If you didn't know, we're very diverse in this area. We have a lot of water in the southeast. Alabama especially. We have 43% of native freshwater gill-breathing snails, and this is in the nation, so or North America. 60% of native mussels, 52% of native freshwater turtles, and we have 306 native freshwater fish species, and that's within the political boundaries of Alabama. That's 38% of the population of fish out of 800 of total in North America. This accounts for 38% are found in Alabama. So the Coosa River Basin, that's what the Choccolocco drains into. 280 miles long, originates in eastern Tennessee, northwestern Georgia, and flows into the Alabama River around Montgomery. Basically covers an area of 10,000 square miles, 147 species of fish, and 10 of these are the greatest conservation needs species, and that's them listed there. It's been said that for comparable drainage to the Coosa River as far as size, the Coosa River is more diverse than any in the world. So 147 species out of that 306 is roughly 48%. So nearly half of all species that are in Alabama are found in this Coosa River. So now this is our little section, this little creek that's just up the road, Choccolocco Creek, Watershed. It drains the area of 507 square miles. Starts in Talladega National Forest and drains into the Coosa River at Logan Martin, right there at Riverside or Lincoln area. It drains 4 counties, Cleveland, Calhoun, Talladega, and Clay. So there's varying opinions on the diversity within Choccolocco Creek. 70 fish species is what it's a Fishes of Alabama book put out written by Bosching Jr. and Meade. I went through and flipped through, and anywhere that had a little dot on Choccolocco Creek where they have found fish, I wrote it down. I counted up 70 species. So that's among most of the professional ichthyologists in the state, that's a pretty reasonable number. 80 species by Paul Johnson, Department of Conservation and Natural Resources. He sent us an email whenever he found out we were doing this little project, and he sent us an email and had 80-something species, but we went through and marked out a good many that he had that were in the wrong drainage. And then there was a study done by the GSA from 97 to 01, and it come up with 61 species. Now this study wasn't done to find species. This study was done to just assess the quality of the stream. So he did miss some that we know are there, and he found some that we don't find. So you can't ever find every species in there, but So that was a pretty good number, but there is definitely some that we know he missed as well. And in this stream system, we find 4 GCN species, those greatest conservation needs. So if you take 70, which is, like I say, a pretty acceptable number, put that into the 306 in Alabama, that's 22%. So nearly a quarter of the species of fish in Alabama are found in Choccolocco Creek. That's 9% in the nation. Out of that 800 species of fish, that 70 is roughly 9%. It's like 8.75%. All right. So that little red splotch right there, that's Choccolocco Creek water drainage. So within Alabama, which is, you know the state outline, Alabama's area is 52,000 square miles. Choccolocco Creek's drainage area is 507. A little bit of math, boom, you find out that less than 1% is what Choccolocco makes up for Alabama's area. So quarter of the fish species are found in less than 1% area of Alabama. That's impressive. All right, so that's kind of a big deal. That's why Choccolocco Creek is so important for these studies, and that's why we have taken this on. Even with all the pollution and everything that we know that's wrong with the system, it still has its high diversity. So our objectives, what is the purpose of this? Develop a current species list. There's not been one put out really recently, so that was kind of thrown in there as something that needs to be done. Survey fish communities, and with these fish communities we can assess the impairment along the stream system. And then also, as you're doing this, you're going to determine ranges for these GCN species, which was another important thing that was needed to be done. So methods. Alabama Comprehensive Wildlife Conservation Strategy. This was put out by the Department of Conservation and Natural Resources. It's the fish chapter, which is chapter 8. I know that thing by heart. It's like 150 pages long. They go through every system in Alabama and tell about the mussels, the snails, the fish, the salamanders, the turtles. They go through everything that is in danger and tell you how it should be assessed, what you need to do, and it tells you that it lists watersheds that are on this very important, need to be studied, and Choccolocco Creek is on that list. And they suggest every 3 to 5 years this needs to have a complete study of the system. All right, so we use backpack electroshocking, seine netting, and snorkeling to do these to find these fish. The EPA has issued a rapid bioassessment protocol for fish surveys, and all this does is this kind of gives you a standard for all these different agencies that are doing this type of work. It gives them a standard to go by, so we're all on the same page. Also, this standard is made to get more bang per buck. You need less people doing more work, so you get more data for your money is the idea. And this, as they've come up with ways to run more efficiently, and that's key with the way money has been hard to come by here lately. So also the Geological Survey of Alabama, they come up with a 30+2 method, and this is your cookie cutter is what I always call it. This makes sure every time you go to the stream A or stream B, you take that cookie cutter and you do a sample, and you take the same cookie cutter over here and do a sample. That way you can compare them. 30+2 means there's 10 pools, 10 runs, and 10 riffles, and 2 banks. So you have 10 riffle habitats that you survey, 10 pool habitats that you survey, and 10 runs, and then 2 banks. And so, like I said, that allows you, that makes sure that you're not surveying 20 riffles here and then come over here and you don't do any riffles and you're doing all pools. You're not going to catch the same fish. You're not going to have that standard. So that was very important that we made sure we had to follow by. And so the Index of Biotic Integrity. This was developed back in '81 by a guy named Carr. He came up with this new idea. It's a little bit different than the Jacquards and the Simpsons and all those different indices. And the Index of Biotic Integrity, you take the fish communities and you can assess the stream by those communities. And he developed a way, and it's a whole long deal of going into that. But anyway, so he suggested that the professionals in the field take this, because he was up in Missouri when he did this. He suggested the professionals take this and implement it in their state or in their region, but to modify it, because Missouri is completely different than Alabama or anywhere else that you go. So O'Neill and the GSA modified it, calibrated it for the Coosa River first in '06, and then they recalibrated it for the whole Alabama Valley and Ridge ecoregion in 2011. And that is the most current, up-to-date IBI that we have out there for this area. And this was developed specifically for this area. So it's a lot better than it used to be. And here's a few sites. There's actually about 4 more since I built this map. There's about 4 more added to it. Just in the past month and a half, I think we've gone out and surveyed a bunch more. So I'm going to break it down into different groups. We're going to look at this upper section first. As you can see, we've done a lot of surveying up there. When Dr. Meade first started with his other graduate students, they were looking at the upper area more, so we have a lot more data for that area. And then recently we have branched down to further along the stream. So this upper area, Upper Choccolocco Creek, high gradient streams, they flow fairly quickly, predominantly riffles and runs. There's not a whole lot of pools or standing water. The land usage in that area is agriculture and natural forest, and these are our most pristine sites. These are some of the best sites that we have, especially in that natural forest, which is, quotations, unimpacted to an extent. It's just actually lesser impacted than the rest of the system. So we're going to look at a site that we surveyed, Brown Bridge Road. There we go. Brown Bridge Road, you can see the IBI score is 40, fair. We'll go into those a little bit later and what they mean exactly. And a lot of this is jargon to people that don't know the fish names. So I broke it down into 3 groups. The green is minnow species, the red is sunfish, and the blue is darters. And the minnows and darters are a little bit more susceptible to habitat changes and water chemistry and such things as that. The sunfish are usually more tolerant. They can live through anything. You go to any mud hole and it's going to have a sunfish in it. So that's kind of what we were looking at, just to kind of get a general idea of what this was saying, was that you can see the changes in these families as you go down the system. So you can see we have a good bit of minnows. Choccolocco Creek normally has a lot of minnows in it. Around 60% of all the fish we catch are in the minnow family. And then you have sunfish and darters there as well. Alright, so that didn't come up like it was supposed to. There we go. Alright, so in this upper system, Shoal Creek is a major tributary, and that's what's circled in red there. And Shoal Creek differs a good bit. It is almost entirely in the National Forest. And so we're going to look separately at Pine Glen Campground here in just a little bit, and that's its location. But a little bit more on Shoal Creek. High gradient streams that originate in Talladega National Forest, tributary to Choccolocco Creek. It was once a site for of 3 GCN species, the blue shiner, coldwater darter, and holiday darter. And this historical data goes back to like the 60s and 70s. So those species haven't been seen since the 80s, I know, in that area. Diverse darter assemblages. Nearly all of the darters that we catch, there's like 8 species of darters. All of them but the coldwater darter we catch in the Shoal Creek area. This is the type locale for the holiday darter. This is the only place you find the holiday darter in Alabama. So it's a very important system based on just the way it is. It's a lot of riffles in that area. It's a really nice system. And that's a holiday darter you see there. So Pine Glen, like I say, this is Pine Glen Campgrounds off there to the left. It's in National Forest. And we're going to break it down into those groups again. You see how many minnows we have, or not minnows, but darters we have there. We have a lot more darters. There are actually 2 that we didn't catch that day, evidently, that aren't in that list. So we catch them there, though. So there's a lot of darters in this area. And like I said, darters are benthic species. They're very susceptible to habitat changes. You start getting sedimentation in, or changes in flow or something like that, you start losing your darters. So they're a key to this IBI scoring. You see, you still have a good bit of minnows, and they both outnumber the sunfish in percent composition and just in numbers of species. So next, we're going to look at the middle reaches of the system. The middle Choccolocco Creek is Oxford-Anniston area. urban land usage, high sedimentation, highly channelized flow with few riffles, and a poor riparian zone. And the riparian zone is really key too. That gives, it not only does it give some habitat on the banks to any debris and also stuff that falls into the creek, but it also shades for temperature changes too. And so that poor riparian zone is another key, as along with the sedimentation. So one of our middle reaches sites is Prees Mill Road. It's kind of a back road close to where I used to live, from Coldwater to Montford. Prees Mill Road, it scored very poor. As you can see, it's a much wider stream than in the other pictures. It's mostly one long pool or one long run, not a whole lot of flow. There is some rocks and stuff, but there's a lot of sedimentation. All the rocks have a little bit of silty covering over them. So we break it down in those two groups. You see, we're missing all darter species from this site. There might be some there, but at this point in time we didn't catch any. But look how many sunfish we have. Those in the red, you have a lot more sunfish. They were the majority of what you found were sunfish there. They had a lot higher composition at that time. And then also, you have only 3 minnow species. So that was another key thing. You went from 60-something% of minnows in the upper region, and then Shoal Creek to like 27% in the mid to lower regions. So you lost a lot of minnows. So Choccolocco Creek watershed again, the lower reaches this time. The lower reaches are very similar to the upper. It's close to the Logan Martin Lake, so you're starting to get into the backwater of the lake itself. It's a lot of agricultural land use. It's long runs, and there's a few shoals in there too, but it's kind of similar to the middle, as where you can see there's just really long runs, and then there'll be a little riffle every several hundred meters, and there'll be a little riffle, and then they'll go for another long run. So there's not a lot of habitat for these darters and minnows. So we're going to look at Jackson Shoals. It's kind of unique for this area. Jackson Shoals is, on either side of it's these long runs like I was talking about, but there is a lot of aquatic vegetation, Jackson Shoals, big riffles in that area. There's not much sedimentation, so there's a lot of habitat. And when you get that habitat, you get your darters back. And there's a good bit of darters in that area on the riffles and everything. They can get in this aquatic vegetation, a lot of them like that. The minnows are the back as well. And you have your sunfish here as well, but they're not in the numbers that they were before. And I'm sure that if you go just a little bit down and you get into that backwater, you're going to start seeing more sunfish, and you're not going to find any darters, and you're not going to find as many minnows. So our results. Jacksonville State University from 2008 to 2012. As you can see, October of 12, we're still doing surveys, including in this data. We surveyed 38 sites on Choccolocco Creek, and we surveyed those sites like 56 or 57 times. So some of them we hit multiple times. We caught 6,700 species of, or not species, 6,700 individuals, and that accounted for 51 species. So you say, all right, you said 70 species earlier, you only caught 51. What are these, why are you having so many missing species? So we come up with 4 reasons. First reason is what a lot of people jump to is they're extirpated. So you have these, as we go along, we lose fish from this area. That's common, or not common occurrence, I wouldn't say, but it happens. And so that could be one possibility, that we have lost species that were once here. But also small populations of species. The GSA, with that survey they did, they caught 11,000 species of fish. Or not species, I keep misstating that. 11,000 fish. And there was a couple of species, they only caught 1 or 2 in that 11,000. That's an extremely small population for one or two fish over a 5-year study. And so that's a small population. That population is going to be hard to detect if it is there. So you can't, you just can't jump out and find, 2 fish and wherever they found on the map. So it makes it difficult to find those small populations. But lake species, this is probably our best bet on why we're a little bit under. we don't have the materials, we don't have the boat shocker or other stuff like that to survey these lakes and these deeper water. We're wadable streams strictly, that's all we survey. So anything that's about chest high, that's all we can survey. So these lake species that like the deeper water, that don't mind the warmer water of the lakes, we've accounted for about 9 species that would be, that we are pretty sure that are there based on fishermen and ourselves fishing and stuff like that. So that would put us up around 60, which is pretty close. And then misidentification. All right, we know this happens with the high diversity that we have. It is easier to misidentify some species, but it doesn't happen so much that you can't use our research, because it is, most of them we do know, we're sure, we try our best to not mis-ID anything. We take it back if we don't know it, we voucher it, and we do our best. But it does happen. Some of these species are very similar. You get hybrids that look like both of its parents, so you really don't know. So calculate IBIs. So I broke this down into 3 sections. First is going to be the upper. That's going to be Shoal Creek, and that's going to be the middle and lowers together. And so all those are just numbers to most of you, so I broke it down. Blue is good. There were no excellent. I'll get to that here in a bit. White is fair, and red is going to be poor or very poor. And as you can see, all the sites in the middle and lower were poor or very poor. Shoal Creek, which was predominantly in that national forest, most of the sites were good or fair, with only 1 scoring poor, and that one's actually a tributary in. So, and then the upper reaches, you can see that most of them scored fair, but there were a good many that scored poor and a good many, or a handful that scored good. So here's a graph. Some people like graphs. This kind of shows you just what I stated. As you see, there are none that are excellent. Section 1 is the upper, Section 2 is shoal, and Section 3 is the middle and lower. There is very few sites that are undisturbed in the nation, if any. And Choccolocco Creek is definitely not one of those places that is undisturbed. So it's hard to find an excellent. Carr stated that it was hard to find an excellent. O'Neill stated that it was hard to find an excellent. And so, some of them are very close. We had 2 sites that were on the border. They had 1 or 2 more points, and they could have scored excellent, but under this survey, we did not. So, go through the IBIs. Next, we're going to talk about the ranges of the GCN species. This is the blue shiner. There's a picture of the little guy right there. Historical data says that 29.6 kilometer section of Choccolocco Creek, which is from that dot there to that dot there. That is the historical range that I could come up with through all my research. Some of them had less, but that was kind of the most up-to-date one that I could find. The GSA did a survey and found a site right here. This is Highway 78 bridge where it crosses Choccolocco Creek. So we're like, hey, we'll go back and we'll survey that site. We'll see if we can catch some blue shiners. We went up there about 15 minutes into it, boom, blue shiners. So we're sure there's a stable population there. So we've actually expanded the range in the current literature in the Fishes of Alabama book. We've expanded the range a little bit. So it should be actually a little bit more than 29.6 kilometers. Also, Kings Gap Branch, that's that site up there. The historical data said there was some there. We went out there, jumped out, boom, blue shiner. So that was February 2012. So we're pretty sure that's a stable population as well. And then Shoal Creek, this is where Shoal Creek comes in, Choccolocco. We hiked down there one day, found blue shiners there as well. That was in historical data. So all these places in historical data, we're finding these fish. So we're sure that there are stable populations here, and that's good. At one point in time, it was inconclusive if they were going to be, if they were stable or not. So Holiday Darter, I already showed you a picture of them earlier. So limited to Shoal Creek, like I said, that's the only place in Alabama they're found. Populations are separated by impoundments. Each one of these dots is separated from the other by a lake. The High Rock Lake didn't show up because it's fairly narrow, but High Rock Lake is like a U-shaped right there. So there's no genetic flow in between these two. You know, they're isolated from each other, so that really affects it. And the fact that if one population gets wiped out for whatever reason, logging or whatever else they do up there, it's exceptionally hard or impossible for them to recolonize this area due to those impoundments. There's no migration in between these, so that was a big issue with these guys. And so they're stable. We find them where, you know, we go back year after year and they're there, but it should be a great concern to the people in that area and what they do, so we can keep those populations. Flame chub, this guy, he's kind of neat. He's found pretty often up in the Tennessee River. He's only found in a few places in this area. We found him here at Joseph Spring. This is the only place in this upper section. We haven't found him in the lower section where they've said they've located him, so I didn't include that yet. But living at Joseph Spring, he's an obligate to spring heads. He can't live anywhere else but these springs, these cold water springs. So if anything happens to the spring head, boom, he's gone. He's out of there, and their populations probably wouldn't make it. So these spring heads are very important. There's possibility that we could find them in other places. We just have to really get out and look more in these springs instead of the main stem. Cold water darter. We located one once back in 2009 up here at Joseph Springs Motorway. Populations exist in Coldwater Spring, which is here. We don't survey Coldwater Spring. There's another fish, the pygmy sculpin. He's next up. I'll show you a little bit about him. These guys are exceptionally endangered. They're on the highest of the highest as far as fish. They're especially in Choccolocco Creek. They are limited to, you know, this one area. So not to get into too much about them, but so we're not allowed up there, federally threatened. So we don't go up there. So we can't find these guys in there either. So that kind of limits us. But we did find one up in this area, like I said, in 2009. Since then we haven't found it. So we don't know if it was an outlier or what happened. And that's the picture of him right there, obviously. So the pygmy sculpin went into a lot. Like I said, headwater, Coldwater Spring. Actually went snorkeling and found this guy, took a picture of him. Little bitty old guy. And it's estimated to be just a few thousand fish. This is the only place in the world this fish lives. It's not that, it's a big stream, but where his habitat in the stream is very small. So Anniston Waterworks owns the stream, or the spring head, and they've made an agreement with Fish and Wildlife that they won't draw out more than 18 million gallons of water a day, and it puts out 32 million. And they have plans implemented that if something were to happen, a catastrophic event at that pumping station, there's a catching method that they go out, scoop up a bunch of them, throw them in tanks to try to preserve them because they're that endangered. Also, there's a few people trying to work in propagation of these guys and to reintroduce them to other maybe suitable spring heads as well. So with that, I'll take any questions. Hopefully I've answered most of them. Yes, sir.
Kind of curious, when you're doing sampling in Middle Choccolocco Creek, do you notice a difference in IDI, species distribution, being sampled a little bit above where Snow Creek with all the PCBs and stuff, versus a little bit below? Was there a really big difference in the sites closest where Snow Creek came in?
Snow Creek's actually one of the sites we've got two right here. Snow Creek confluence with Choccolocco Creek and Snow Creek at O'Charlie's. We don't have a site right above that. Let's see, what's the next one? Near Cider Ridge. That's a pretty good ways above Snow Creek, and it's still very poor. So I would not imagine that area, not necessarily isn't affected by PCBs, but that whole area of the middle reaches is pretty affected. And it's more habitat degradation than anything. The sedimentation kills off these little insects, and then that kills off the other animals that are in that area. So so it's not that the water quality doesn't matter, it's but that whole reach is all pretty bad off. So, but no, not to answer your question. We haven't surveyed just right above it to see if there was difference than right below. That that might be something to look at in the future. Yes, sir.
You run into any exotics?
We find western mosquito fish. It is considered naturalized for the most part, but it is exotic. We find them pretty regular. I mean, they're not coming in and wiping out massive species, but
So those loaches are not in this area yet?
not yet. They're in the same kind of slew of Logan Martin, but in the lower areas, and they could be in there a little bit further down than Jackson Shoal. So, like I say, they're not quite that far up yet. Yes, sir.
How do you define IBI? I missed this.
Index of biotic integrity.
Yeah.
Just
to get this number, there must be some formula.
Yeah, there's 12 different metrics that go into it, and it's anything from the number of species that you have that you catch in this area, and the number of the darter species that you catch, number of minnow species that you catch, the percent of them that are omnivores versus the percent of them that are carnivores. And there's a paper, and it's that thick article or a report that was given out by whenever he calibrated this. And it's all based on the size of the watershed from that point up. So it's really hard to quantify and to tell because it's on the log scale and all this other crazy stuff. But they went through and they looked at habitat degradation versus what they found at this site, and then they did statistically correlated it with each other, and that's how they determined what to add. But it's based on the fish, based on feeding guilds that the fish have, and then percentage of what they find. So it's a little bit difficult to describe. Yes, sir.
Just related to that, does it include the abundances of the fish or just presence or absence?
Yeah, it does. There are some percent. Usually the feeding guilds are percent. It's percent top carnivores or percent omnivores, herbivores.
No, I mean like abundances. An individual, like say you caught 20 of these darters or
No.
sunfish, does it take into account the numbers of the individuals of the species?
No. It's
Okay.
we call it 8
basically
species of
presence or
darters.
absence, okay.
Yeah, it's
So on the species level, it's basically presence or absence.
yeah, usually just on the species level. Yeah, it's about 50-50. Half of it's based on percentage abundance, but that's usually feeding guilds or breeding guilds if it's a open water breather or if it's a substrate breeder. Did you raise your hand? You sure you don't have a question?
No, not yet.
Anything else? I know Dr. TJ has.
Oh, yeah, I can't help it. What is historical data?
Historical data, there's, it's hard to say. Fishes of Alabama books, they go through and they read dozens of articles about the whole area, about all of Alabama, but they have read articles, and then I go back and reread their articles they said they read, and then I try to find my own. Let's see, Kuhada has done a lot of stuff in this area. Meade has done a lot of stuff in this area. It's just articles they've produced. There's been several reports given by Carol Johnson through Auburn. She gives reports to the National Forest, and O'Neill and the GSA. I use their stuff a lot for historical data as well. And like I said, it dates back. I got some papers that date back to the 60s. And I wanted to get that old data because that's before we had the impoundments put in on Shoal Creek. Yes, sir.
For me, actually, I know nothing about this, first of all, let me say that. But it looks like that those fishes are very small, so they have no commercial value.
No, well, all the GCN species are small. In this case, not all of them in general. Let's see. The sunfish, they're popular, and the bass. Sunfish bass are sunfish, but they're popular on recreational sport fishing. But as far as we don't have salmon or anything like that, or that. So.
But why don't you have some big fishes here? You didn't catch them, or they were not? You simply were electrocuting with too small voltage.
Oh no, we zap them pretty good.
Okay, so,
No, what
so they escape.
Yeah, we have poor netmen, I guess. But no, we do catch big fish. Some of the suckers are very large. We caught a sucker fish that was probably 12, or it's getting bigger every time I tell the story. But it was, you know, it was 12 inches or so. We catch several that are big. The brim can be pretty big. But usually your big species are going to be those big lake species that we don't survey for. Your big bass, the bass that we normally catch are fairly small. It's the juveniles that are growing up in these shallower areas, and then they migrate into deeper water. There are big fish. We went snorkeling below one of the reservoirs, and there was bass that were, I feel like they were as big as me when you're in the water with them. But they were, you know, well over 10 or 15 pounds. So we do have big fish in there. We just don't catch them in these wadable streams that often. Yes, sir.
Did you run any comparisons, any statistics on comparing your IBI scores from your headwaters, your middle area, and your
No, I did not. Since most of the time we just surveyed a lot of them once, it wasn't really going to give us that much to go on. And so that I did do a little bit based on the GSA survey, but that was also hard to do because we didn't have we had their average IBI. So they went out 5 times or 8 times and surveyed this site, and they just gave us the average and a range. So we really didn't know their numbers to get to run all the standard deviations and the t-test and all that. So, but no, to answer your question, no, we did not.
I just was curious. I guess what I was thinking was those 38 sites, those are the ones that you did, right?
Yes. Yes, sir.
When you're looking at them, you see your scores varying a little bit, and I know you did it looked like you did this over 4 years is what I'm looking at.
Yes, sir.
And I presume they have to be done at different times of the year too. You couldn't have gone out and done all of them at the same time. I was wondering if, you know, maybe for your upper and your middle reaches, if you look at the time of the year, what year, and what time of the year, whether the scores would correlate with that or not. Yes. I don't know.
Yes, there's definitely going to be some difference in seasonality with a project like this. It would, you know, usually they just go out and they go survey. You know, if you want to base it off seasonality, you know, that would be something reasonable to do. But
that's not what I mean. What I mean is like in your one reach here where you got Kathy's Creek being poor.
Yes.
Is it poor because Kathy Creek is poor, or is it poor because that was the time of year that you did it? So if you sort of plotted them by time of year, you might be able to say, oh, okay, this is why Kathy's Creek turned out this way. Or maybe that might show up more in the middle reaches where you got more of a mixed bag.
Yes, sir.
So, I mean, I don't think anything would happen with this site over here.
Yeah. Kathy's Creek, that specifically is a tributary.
Yeah.
It's above, it drains directly into an impoundment, and it's a headwater spring. There's not much habitat, large boulders, stuff like that. So I would feel comfortable in saying no, Kathy's Creek would not. But that's also Dry Creek, Conaquilla, and Joseph Spring. They're all headwaters as well, and the headwaters sometimes struggle to do it as well because the habitat's not there.
Okay. Since they're closed systems, they can't migrate out of those systems. So would you expect them to change populations or anything in the winter versus the summer? Or,
well, whenever you look at a stream that's a couple kilometers long and you're taking 150 meters out of it, or not even that far, or like 100 meters sometimes, whenever you look at that small of an area, you could have populations migrate from the lower or the upper based on, you know, that you're taking a small sample out of that area. But probably I wouldn't expect to see much migration in that. Most of these species, especially the minnows, there were studies done on the blue shiner itself, and they don't migrate very far at all. So I wouldn't expect them to, but still you are taking a small section of a very large system. Yes, sir.
I'm probably asking too many questions, but what criteria did you use to basically divide this into 3 groups, right? Actually, because for me, like 34 points looks like 35, very much the same. So why this 34 means, well, needs in greater need of conservation than say 35?
To be honest, they both need it but that's the standard that was set up by the people that developed the
Someone
IVI.
basically set up like A, B, C, D, F, right?
Pretty much.
Yeah, that's what that big paper he was talking about, that big IVI
Yeah.
paper. They did all that correlation, said this is a good habitat, this is a moderate habitat, bad habitat, and when you do these scores, that's what those numbers are.
So does this index depend on the time of year, supposedly?
Well,
Yeah.
that's what I was just alluding. I'm not sure.
You
I
will
guess,
have
I guess
some-
it could. I guess.
Yeah, you will have some variation, but a bad habitat's a bad habitat, and it's not going to have good populations regardless of time of year. That's usually the thinking. You might have one jump if it's borderline poor. It might jump to borderline or just over to fair, or vice versa. But you're not going to have one that's poor go to good because it's springtime instead of fall. So